Welcome to the Scientific Sense podcast, where we explore emerging ideas from science, policy, economics, and technology. My name is Gil Eapen. We talk with world's leading academics and experts about their recent research or general areas of topical interest. Scientific Sense is an unstructured conversation with no agenda or preparation. We cover a wide variety of domains where new discoveries are made and new technologies are developed on a daily basis. We are most interested in how new ideas affect society and help educate the world how to pursue a rewarding and enjoyable life rooted in science, logic, and information. We seek knowledge without boundaries or constraints and provide unedited content of conversations with researchers and leaders who love what they do. A companion blog to this podcast can be found at scientificsense.com, and this podcast is available on over a dozen platforms and directly at scientificsense.net. If you have suggestions for topics, guests, and other ideas, please send them to info@scientificsense.com, and I can be reached at gil@eapen.info. My guest today is Professor James Bashkin, who is a professor of chemistry and biochemistry at the University of Missouri, St. Louis. Before, he was an NIH postdoctoral fellow at Harvard, before joining Monsanto Corporate Research, which later became Pharmacia and then Pfizer. His recent research interest has been at the interface of chemistry and biology in areas such as chemical genomics, the design of antiviral and anticancer agents, and green chemistry. Welcome, Jim.
Thanks very much for having me, Gil.
I want to start with a topical idea that you're pursuing, and the project you have is inexpensive sterilization of PPE for the COVID-19 pandemic. And you say that you will make inexpensive, portable, reagent-free sterilizers for N95 masks and other personal protective equipment. Could you talk a little bit about that project?
Well, that's a collaboration with KWJ Engineering and Spec Sensors.
Yeah.
And we're still waiting for funding on that project. And I guess I'm not sure.
We can get into details, okay.
I'm at liberty to say about that, except that KWJ and Spec Sensors have a great deal of expertise in electrochemical detection, and that turns out to be very useful for making a 12-volt based decontamination chamber that can be anywhere from 5 gallons on up in size. And so the lowest size would cost around $300 and would be able to sterilize on the order of 10 or more masks
at one time.
At one time,
yeah.
Right. And so it's the kind of thing that any doctor's office or ambulance or police station or fire station could have. The current sterilization equipment that's available is, for the most part, not entirely, but for the most part, very expensive and limited to large hospitals where contaminated masks have to be collected and brought to a centralized location. So we're hoping that this would be a way to bring sterilization of equipment to a much broader audience, and hopefully also to developing countries where things could be run with solar power and that sort of thing.
Yeah. Yeah, I mean, that's, one of the issues obviously we ran into is the availability of N95 masks early on and the lack of PPE.
Right.
And seems like that was sort of taken care of till we thought numbers are going down, but now numbers are exponentially rising again. So I think this will be a big issue, um, not only in kind of wave one that we are in, but also potentially a wave two that might be coming behind it, right?
Right. Exactly. And the I mean, I was in the hospital for a necessary surgical procedure while in the early stages of phase one, and the nurses there were reusing surgical masks for 3 shifts in a row,
Hmm.
and apparently they had to militate to even get access to those masks, and several nurses quit because they weren't being given masks. One of the local hospitals until they finally, until policies were finally changed. And that was pretty early on in the process. But as you say, supplies became less limited, but now problems are arising again with all kinds of things from the test kit supply chain to PPE.
Yeah, and if the price range for something like this sort of distributed sterilizing equipment, that's going to be a great help for developing countries, right? Brazil is pushing on 2 million cases. India is reaching 1 million cases, and doesn't appear to be either one of these countries have hit the peak yet.
Absolutely, and neither have we.
Right, right. But let's jump into some of your major research areas and interests. So your main research, Jim, correct me if I'm wrong, involves the chemical synthesis and biochemical testing of sequence-specific DNA binding molecules designed to control gene expression. And more recently, you have been working toward prevention of cervical cancer. Now, cervical cancer, most types, is caused by human papillomavirus, HPV 16 and 18, and some of the agents that your team has designed successfully eliminate HPV 16 DNA from human cells in culture. So this is essentially a cure for somebody who has the problem.
Well, it's not really a cure, but these are designed as antiviral drugs that would be treatments for the disease. And
Okay.
so and all or almost all antiviral drugs are designed to control the disease that they affect rather than cure the disease. There aren't very many cases where cures are actually affected, although they occasionally happen.
Yeah.
But the point is that not very many people are being vaccinated worldwide, and even in this country, the vaccination numbers are not as high as we would like them to be. And furthermore, in addition to HPV 16 and 18, there are at least 15 total types of HPV that are recognized as high risk and that cause cervical and other cancers. And HPV 16 and 18 account for about 70% of cervical cancer, but the remaining 30% are caused by HPV 31 and other so-called minor high-risk forms of HPV, but they're not minor to the people who are infected with them.
Sure, yeah.
And so for patients who have infections, it would be nice to have a drug therapy that can treat those infections it's been shown in the clinic that the progress towards cervical cancer is linked to the amount of HPV-16 in somebody's system, or the viral titer.
Yeah.
And so if we extrapolate from that and say that's the case for the other high-risk HPVs, if we can drop the viral titer down to a very low level with an antiviral drug, then we have the chance of greatly limiting the and maybe completely slowing the progress to cancer for these precancerous patients.
Right. Correct me if I'm wrong, Jim. So HPV, there are vaccines for HPV, both a male and female variety. And as you say, it's not widely or broadly applied. And because of that, people get infected with HPV. And the hypothesis, or if it is completely known, that if HPV is in the system for a long period of time, it can lead to cervical cancer, right? Is that the idea?
Yes, that's right.
Okay.
And so, HPV infections are extremely common, but only about 10% of those infections remain as persistent infections, or maybe even less than 10% remain as persistent infections. Many people clear the infections with their immune responses to the virus.
Yeah.
And unfortunately, not everybody does, through some combination of their immune system and the particular virus, the version of the virus they've been infected with.
Right.
But in addition, HPV has been showing up in a wide range of other cancers, including head and neck cancers, such as oral cancers. And so there's a really vast number of increasing head and neck cancer cases directly in which HPV-16 is directly implicated. And so we don't have a nice testing system like the Pap smear that we for oral cases that's been developed. Although the Pap smear is not a perfect test by any means, it saves a lot of lives and has been responsible for a decrease in the death rate in developed countries due to cervical cancer.
Right. And there have been different conjectures, right? So cancer is caused by virus, cancer is caused by chemicals or genetic abnormalities or mutations. What is the dominant position now, looking at cancers more broadly? What do we believe cancer is caused by?
Well, I think it's all of the above.
Okay.
And all of the above contribute to all of the things you mentioned contribute to risk factors.
Yeah.
And the more risk factors one accumulates, the higher the risk one has of developing cancer. But certain risk factors are particularly serious, and high risk HPV infections are among those that can lead very direct. Well, that can and do lead very directly to, or relatively directly to cancer.
Yeah. And so the antiviral agents that you have designed, if it is able to completely eliminate HPV-16, somebody who hasn't developed any cancer yet, if it is able to completely eliminate HPV-16, then that individual's risk of a virus-driven cancer could be substantially eliminated.
Yes. Now, we've never seen 100% elimination, but we've seen many, many fold elimination. So I should point out that we did not do the virology for this project ourselves. The virology was done by Chris Fisher and Terry Edwards.
Yeah.
And they made all they did all of the science related to measuring the effectiveness of the compounds, and Chris Fisher was heavily involved in designing the entire project with me from the very start. But what we see is that the viral genome of HPV, whether it's 16 or 18 or 31, is destroyed, is cut by human enzymes after our compounds are added to cells that contain the circular viral genome of DNA. Our compounds don't have any effect on cells once HPV has integrated into the host cells. So that's part of the life cycle of HPV, that the circular DNA opens up to a linear piece and inserts into human DNA. But that can take 15 to 20 years to happen. And so we have a window of time in which antiviral therapy is appropriate. And that's when our compounds or any other anti-HPV antiviral would be effective.
And how long are these therapies typically?
Well,
depends.
Our work has been done in cell culture and tissue culture. Yeah. And so in 48 hours, we see decreases of as much as 99% of the amount of viral DNA with a single dose of compound.
So these will be going into the clinic in the future?
Well, I hope so, but we need to obtain the funding to scale up to do the last of the preclinical studies, and then try to file an investigational new drug application.
Okay. And all these agents, I know, Jim, you have a particular focus on polyamides. So all of these agents are antiviral polyamides?
No, actually, there's a selection of sorts. So some of the polyamides are antiviral towards small double-stranded DNA tumor viruses, and they seem to be somewhat broad spectrum in that activity. But they have to be for the most part, they have to be relatively large molecules. And we've seen antiviral activity against a negative strand RNA virus related to respiratory syncytial virus with a smaller polyamide. But the small polyamides have not typically been active against HPV or other double-strand DNA tumor viruses. And the large molecules, which have, say, 14 rings in them or more, are fairly high in molecular weight, and a lot of traditional medicinal chemists think that they would never get into cells. But we found that there's an active uptake mechanism for them with at least many cell types, including the cells that the human skin cells that are infected by HPV. And so they get in quite effectively to the cells that carry HPV infections of the high risk type, and they are therefore able to bind to the viral DNA, which is what we believe is the mechanism of action. Then there's some molecular details that we have yet to work out about how that event, that binding event, triggers the cell response to recognize the viral DNA as foreign and destroy it. Normally, HPV is very good at hiding from the DNA damage response and co-opting the DNA damage response to help the virus replicate in the case of HPV and related viruses.
Is COVID-19 an RNA-based virus?
Yes, it is.
And so it also has applications in single-strand RNA-based viruses too, right?
We have found one example of a compound that's active against an RNA, a single-strand RNA virus. Yes.
Okay. And that's something that you're currently working on, or it's too early to?
We are.
Okay.
We're trying to find additional examples. So one example wasn't enough to encourage the NIH to fund us to look at it in more detail. Although we have our collaborator there is Ming Lo at Georgia State University, and his group was able to crystallize the nucleocapsid-like particle of the virus with RNA, with the RNA-like genome in the center and the nucleocapsid protein particles surrounding that RNA, and our compound bound to that RNA protein complex. And so we can see evidence of the potential mechanism of action from that crystal structure, and also from biophysical studies they did showing that the melting temperature of the particle is increased, or the particle is stabilized by the interactions between our compound and the RNA and the protein. And as far as I know, it's the first example, sorry to interrupt you, it's the first example of one of these polyamides binding to RNA, and also the first example of one binding to protein.
Yeah. And so polyamide, so these are, I would imagine, highly scalable compounds, right? I mean, if you find activity and if you find, to be efficacious from a manufacturing perspective, it shouldn't really create a big issue, right?
Yeah, that's pretty much the case. So we've scaled up 2 compounds to a 10-gram scale, and used those for animal testing for safety studies for preclinical work. And it's making them is a bit like peptide synthesis.
Yeah.
However, there is one major difference. They are based on amino acids like peptides are, but the amino acids used to make the pyrrole imidazole polyamides are, for the most part, heterocyclic amino acids based on nitrogen-containing heterocycles. And I know that's starting to get a little technical, but what it means is that the amino groups of the amino acids don't react very well in the amide coupling reactions compared to, say, the alpha amino groups of amino acids used to make peptides. The alpha amino groups of the common amino acids are very good nucleophiles compared to the amino groups we work with, which are on electron withdrawing heterocycles containing nitrogen.
Yeah.
And so we have to use a lot more excess equivalents. We have of reagents to get good coupling efficiencies.
Oh, yeah.
And so that makes the process of scaling up more complex than it would look like on paper.
Mm-hmm.
Nevertheless, it is a scalable process. And one of the things that we do is make is join two building blocks together in solution beforehand, and then add the building blocks in twos instead of one at a time to improve the efficiency of the solid phase Merrifield type synthesis that we do carry out.
Yeah. I want to jump into another recent paper that you have, Jim. It's about an early form of skin cancer.
Mm-hmm.
Could you talk a little bit about that?
I'm sorry, which?
It's the Bowen's disease.
Yes, that's a patent.
Okay.
And so I'm working with a gentleman named Dr. Justice Obi, who is an MD and a pharmacist, and he works in New York as a pharmacist. And he discovered that chloroquine has anti-HPV activities, and the NIH sponsored tests run at a number of universities to do cell culture and tissue culture assays to see if they could confirm or deny the observations that he'd made anecdotally. And those tests confirmed at the molecular biology and DNA level what he'd observed by eye. And so he has used this medication, which has been in the news quite a lot lately, in off-label treatments for people suffering from a variety of HPV-derived diseases, including Bowen's disease, which is a precancerous and then later cancerous skin condition that's very slow progressing type of cancer. And he treated several people with this condition, and their condition completely cleared up. So he's formulated chloroquine to be used as a mouthwash and as a topical cream and gel for both internal and use in internal vaginal cervical use and anal use, and also external use on genital warts. So we have a patent on that.
Yeah. I remember chloroquine was used sort of an anti-malaria drug, right? It has been around for a long time, isn't it?
Yes, that's right.
So I remember, when you go to tropical areas, you used to take it just as a preventative measure. And now I guess we are finding a lot of uses for it in elsewhere.
Right. So we don't have a complete mechanism of action for it, but it turns out that chloroquine is it's in about 20 clinical trials right now in combination therapy as an anti-cancer drug. Mm-hmm. And one of the effects that it has is to reestablish concentrations of a tumor suppressor protein that it turns out HPV gets rid of.
Yeah.
And so it may be that the tumor suppressor protein is in fact turned on by chloroquine, and this interferes with the life cycle of HPV, and that leads to the loss of viability of the life cycle for HPV. So P53 is the tumor suppressor protein, and one of the main oncogenes of HPV, known as E6, and its gene product, the E6 protein, causes P53 to be ubiquitinated and degraded in the proteasome. So ubiquitin is a molecular signature that's used to tag proteins for degradation. And so there's basically no tumor suppressor protein in HPV-infected cells. And the fact that chloroquine can reestablish P53 synthesis in tumor cells and cause regression of the tumors has led to its use in anticancer trials, and that may be why it's effective against HPV also. But that's speculation. We don't have real data to tell us whether that's the true mechanism.
Yeah, but it's encouraging because the effects appear to be there, right? In HPV driven, some of these HPV driven diseases. I know, Jim, the one other area that you have a lot of interest in is this environmentally benign organic chemistry known as green chemistry.
Yes.
So I know that Pfizer. Folks at Pfizer have been very interested in it when I was there in the late 90s. What's the state of the art in green chemistry now?
Well, the state of the art really is to consider the principles of green chemistry, atom economy, energy economy, waste stream, and to try to consider those principles whenever any manufacturing is taken online or before it's taken online, so that processes are as atom efficient as possible, as energy efficient as possible, and that the generation of harmful waste streams is eliminated or minimized as much as possible.
Yeah.
And that includes minimizing the use of solvents that are harmful, both because they're fire hazards and they're hazardous to the workers, but also because in many cases they end up in water treatment facilities and may or may not be completely dealt with by those water treatment facilities, at least initially. So there are a lot of people who are concerned with this in the commodity chemicals area and the pharmaceutical area, but still probably not nearly enough is put into practice because of the timelines involved in commercializing things. And it's very hard to rethink a synthesis of a pharmaceutical for scale up when one is rushing to make samples for clinical trials.
Yeah.
And so one of the things that I hope is that chemists, organic chemists at the bench will start using greener methods in their daily discovery chemical reactions, so that the syntheses that need to be scaled up will already have green bits built into them, green building blocks built into them when it comes time to scale things up. But I think we're a ways away from that, because expedience seems to win out over other methods in the discovery phase. Everything's about speed at that stage.
Yeah, I mean, at the very highest level, it's sort of a cost minimization problem, right? So I wondered, cost meaning, overall cost of production, all externalized costs. So I wondered if there are computational techniques, maybe some AI type techniques that could be deployed in this area to get some guidance as to what might be optimum.
There are computational techniques that people have developed that are pretty effective, and the fact is that the process I worked on with Mike Stern and Roger Rains and others at Jim Allman at Monsanto and Solutia for rubber chemicals intermediates in the 1990s actually ended up with a much less expensive process than the one it replaced, and eliminated waste dramatically. So that's sort of a textbook case where, some people might say, worrying about the environment always costs industry more money. But it's a case where it actually made industry a lot of money and allowed one company to essentially dominate or at least one company and those companies who had access to the same technology to dominate the globe, and bring down or control the price, and eliminate this 99% of the waste that was associated with the old chemistry. So sometimes it just takes starting from scratch, to looking at a chemical process with a new set of eyes and a new set of priorities. And a lot of the bad actors are chlorobenzenes or other halogenated compounds, because they put a lot of salt into waste streams, and that's ultimately harmful to things that live in aqueous waste streams, to the entire biosphere. And we were able to eliminate that salt and also, cut costs really substantially, because we never had to make the chlorobenzenes in the first place just to take the chlorine off later on. And so it ended up being much less expensive.
Yeah, it's counterintuitive, right? That's why sometimes, people don't go into it. But similar things have been seen in manufacturing, where you design in quality aspects very early in the process. People say, well, it's going to be costly to do so, but ultimately, if you look at the entire process, you actually minimize cost that way if you do it systematically and programmatically. But it's counterintuitive for decision makers. That's the issue.
That's right. And the thing about it, well, it really depends on whether we're talking about commodity chemicals or pharmaceuticals, because the big cost one of the big costs in pharmaceutical research is all of the failed programs.
Yeah.
And as you know, so the speed with which one can test compounds, go through examples, and decide whether they're good or bad and worth moving on with or need to be discarded is a critical factor. Mm-hmm. And so that is the primary motivating factor in governing decisions for pharmaceutical research, at least of certain kinds. But people who people who are looking at commodity chemicals and who may want to retool their plants have to consider the capital costs of retooling a facility versus the environmental savings and ultimate total cost savings. And sometimes it doesn't take a lot of capital improvement to put in a new process, and other times it does. It just depends on the details.
Yeah, I wonder, Jim, I mean, you have a lot of experience inside pharmaceutical companies too. And I wondered, from an organizational perspective, do you think a specialized green chemistry group inside a pharmaceutical company who almost runs things in parallel as the compound progresses, do you think something like that from an organizational perspective would work better?
Well, groups like that do exist. And I know Pfizer has at various times had a group like that. And I think that they can contribute a great deal. So it's just I think the problem is that there can be maybe 20 projects going on, each of which has 20 compounds they're investigating, and then all of a sudden
you just don't have the resources.
Five of those projects move forward to the next stage, each with one compound very quickly, and trying to make an impact in a very short time on how that synthesis occurs is not so easy.
Yeah.
Because changing things at that stage can change the purity of the molecule, the impurity profile, and all kinds of things that are starting to be established. And so, yes, I mean, those groups can have a large role to play, but how often they get to play it varies on the details of each project.
Right. So in conclusion, Jim, if you look forward 4 or 5 years, which area do you think has, from all the research things that you're working on, which area do you think has the maximum potential, to go to the next stage or more interesting discoveries?
Well, I think that both of our HPV-related projects have great potential. I mean, chloroquine has a lot of potential because the drug is extremely cheap and readily available, and we have over 100 anecdotal, probably about 200 at this point, anecdotal patients who have been treated, and we have DNA tests backing up the success of the treatment in a number of cases. But that's not the same as a double-blind clinical trial. So, it really needs to be subjected to a proper clinical trial, and we're trying to raise the funding to do that. And I think that our other HPV compounds, anti-HPV compounds, also have a lot of potential, and they have potential in other areas too, against other double-stranded DNA tumor viruses, such as the polyomaviruses, which are important for organ transplant patients, bone marrow transplant patients, and patients along those lines. So there we might be able to treat bone marrow before it's transplanted into a patient, especially for juvenile bone marrow transplant patients. And I was hoping to get started on looking at that kind of thing when the COVID-19 crisis hit, and everybody became preoccupied with other things.
Yeah, I mean, it's exciting because it sounds like it's a platform that you could take in a lot of different directions.
Yes, I think so. And obviously, we need to do more testing, but that's certainly the hope.
Yeah, yeah. This has been excellent, Jim. Thanks so much for spending time with me, and good luck with all the research.
Thank you very much, Gil. I really enjoyed talking to you.
Thank you. Bye.
Bye-bye.